In this presentation, we'll examine secondary structure of nucleic acids. For determination of secondary structure of DNA, a historically important point is Chargaf's rule. Chargaf was an Austrian biochemist, and well before structure of DNA was known, he came up with his rule. Actually, there is more than one rule, but most important rule states that DNA of any species, of any organism, has 1-to-1 stoichiometric ratio of purine and pyrimidine bases. And more specifically, amounts of adenine and thymine are identical, and amounts of guanine and cytosine are identical. To us, it has some practical importance. It means that, for example, if you know content of just one of the nucleic acid bases, then we know content of all of them. And here is an example of such problem. If molecule of DNA contains 18% adenine, what is its cytosine content? Well, since adenine pairs up with thymine, that means that thymine content must be the same as adenine. It must be 18%. And in turn, that means that adenine and thymine together comprise 36% of the molecule. Rest, 64%, must be guanine-cytosine pairs, and then half of that, or 32%, is cytosine. Chargaf's rule is actually consequence of base pairing. So particular bases pair up in a molecule of DNA or sometimes in segments of double-stranded RNA. And those pairs are cytosine guanine and adenine thymine in DNA. Of course, it would be adenine uracil in RNA. Pairing is consequence of intermolecular hydrogen bonding. And you can see here that there are 3 intermolecular hydrogen bonds in cytosine guanine pair and 2 intermolecular hydrogen bonds in adenine thymine pair. Cytosine guanine pair is usually referred to as GC, or guanine cytosine pair, but here in this formula, because cytosine is shown on the left, then I'm showing it as cytosine guanine pair. Here are the same hydrogen bonds shown as they would appear in nucleotide residues. So here we have nucleotide residues that are parts of DNA chain, and same pairing is shown. Here is yet another representation. So on the left we have 4 base segments of a chain of a DNA, and on the right 2 of the pairings are shown. So even shorter 2 base segments of DNA chain is shown, complementary chain is shown on the right, and a base pairing is shown again. Secondary structure of DNA is consequence of intermolecular interactions. But while base pairing is consequence of hydrogen bonding between complementary bases, secondary structure is actually not consequence of hydrogen bonding. Because if it were simply consequence of hydrogen bonding between individual base pairs, then we could come up with some hypothetical structure which would be reasonably stable, and that's ladder structure. In fact, since DNA exists in aqueous solution, individual bases could make just as strong hydrogen bonds to molecules of water, so there would be actually no reason even for them to pair up. But there are other intermolecular interactions that are involved, and they cause actually bases both to pair up and interact through hydrogen bonding, and also for molecule of DNA to assume particular secondary structure. So ladder structure would account for base pairing, for intermolecular hydrogen bond interactions between individual base pairs, and of course for the structure of the molecule that contains sugar phosphate backbone, shown here in blue, and individual bases, here shown in different colors. But this structure has one major weakness, and it is that sides of each base pair would be exposed to water. Those sides are hydrophobic, so those would be unfavorable hydrophobic interactions, because hydrogen bonds between molecules of water would be broken, and they would be replaced by weak dipole-induced dipole, or basically weaker dipole-dipole interactions. To avoid such hydrophobic interactions, this hypothetical ladder structure twists into a double helix. This double helix retains regular appropriate distance between individual deoxyribose units in the backbone, but it excludes water molecules. It basically squeezes out water molecules and makes sure that only individual base pairs interact with each other through dipole-dipole or basically hydrophobic interactions. Here is again double helix of DNA, and we can examine it in more detail. Negatively charged phosphate groups are now all on the exterior surface of the helix, and that way repulsive effects of one another of negative charges of phosphate are minimized. Those phosphate groups or phosphate ions electrostatically shield one another because different cations bind to them, such as magnesium cation, and also they strongly interact with water to form hydrogen bonding. The same way, sugar part of the sugar phosphate backbone interacts with water to form hydrogen bonding. On the other hand, the inside, the core of the helix, consists of base pairs. And those bases are, you can see, stuck up on one on top of the other. They interact through pi-pi electrostatic interactions, which is form of actually van der Waals interactions, which would be intermolecular interactions, or more specifically, a form of London interactions. We can identify in each double helix a distance between individual base pairs. And distance between individual base pairs is 0.34 nanometers. The DNA double helix is twisted in such a way that helix repeats itself approximately every 10 base pairs, which means that pitch of the helix is 3.4 nanometers. This is the most common conformation of DNA in solution, and it's called B-DNA. There are also A and Z forms, which we'll not be covering in this presentation. In a double helix of DNA, we can also identify major groove and minor groove. And here is a view of DNA double helix from the top. So sugar phosphate backbone is shown as a red circle. Of course, it's a spiral. Since we view it from the top, it appears as a circle. Glycosidic bond to each of the bases is shown as solid bold bond to the nitrogen, and intermolecular hydrogen bonds are also indicated. So now in the view from the top, you can also identify what major groove and minor groove looks like in each base pair. And you can see each of the three possible base pairs: AT base pair of DNA, AU base pair of RNA, and GC base pair, which is common to DNA and RNA. This completes a review of secondary structure of nucleic acids, and next we'll be looking at structure of eukaryotic chromosomes.